Okay, so today, uh, we have already done quite a bit on the anatomy of ear. We have talked about the external ear, then we talked about the middle ear, and then last session we had quite a bit on the anatomy of the inner ear. So this session is going to be more about the physiology, and so we would dive in more in details about physiology, like rather than the structure. So I think with you guys already knowing the structure, it would be a little bit much more easier, a little bit of recap of things which you have learned in anatomy, but a little bit of more deep dive into the hearing mechanism. One thing which we are going to do a little bit in more detail today is more of the function of the inner ear and how what part of your hearing is basically analyzed at the level of the organ of Porta, and what happens, in the central hearing mechanism, which parts are responsible for localization, intensity. So we would have a little bit of more detailed discussion today about the hearing pathway. That is also something which we are going to do a little bit more on detail, and then we are going to talk about the different types of deafness, and then how we can do simple tests to identify if this is a conductive deafness or sensitive deafness. So what we are going to do today is to describe the function of the outer ear. We are going to talk about the function of the middle ear, inner ear, and listing in order the mechanical structures over which the sound energy is transmitted to the auditory receptors. We will draw a human audibility curve, the ranges of frequency and loudness which we can hear, and we'll compare it with other species and see, where we are basically standing in terms of frequency of the sound and loudness of the sound. We are also going to, talk about the frequency analysis by the cochlea. How does the cochlea is able to identify frequency of different sounds? We are also going to talk about the action potentials generated in the hair cells. What are the mechanisms? We will do a detailed auditory pathway. So this session will have a more detailed auditory pathway. You are going to go all the way up to the cortex, and we will see which central nuclei are responsible for what. We would talk about how pitch, that means the frequency, loudness, localization of sound in space is coded by the central nervous system, and which parts of the central nervous system are responsible for localization of the sounds and the loudness of the sound or intensity of the sound. And then we are going to finally distinguish between conductive and sensorineural deafness and different types of tests which we perform usually to identify different types of deafness. So first we are going to talk about a little bit about sound itself. So sound waves are basically traveling in the air in the form of compressions and rarefactions. So you have compression, rarefaction, compression, rarefaction. This is how the sound waves are conducted in the air. The audible range of frequency of sound is between 20 to 20,000 hertz. This is our audible frequency of the sound which we can hear. And in terms of amplitude or loudness, we can hear up to 80 to 120 decibels. This is the range in which we can maximum hear the sound waves, in terms of loudness. If it is above that, we can't hear it. If it is below that, we can't hear it. So now, if you are going to assess, for example, the sound, usually what you have is a graph. you have a graph which has pressure there on the y-axis, and you have time in the x-axis. So when you see the sound waves, when they are like this, they're going like this. So the peaks are showing the pressure, and that is basically telling you about the amplitude of the sound waves, or the loudness of these sound waves, and how frequently they are coming. These sound waves is what is going to tell you the frequency. So if they are coming close to each other, that means they are high frequency sounds. If they are coming apart, these waves which you are seeing, then this is low frequency sound. And how big is the pressure of the wave is telling you the loudness of the sound. So this is what you need to understand. So our peak sensitivity, that means the highest sensitivity at which you can hear the sound, so that is between 1000 to 4000 hertz. This is, your ears are prone to hear the sound in this frequency range the best. And this is the frequency range which you usually speak. So when we are speaking, our range of frequency of sound is between 250 hertz to 4000 hertz. And so it is coming in between the same range where which is the highest sensitivity or peak sensitivity of our ears to hear the sound. Sound transduction, as we have talked several times before, that it is in the form of airwaves. It passes through the external auditory, or it is funneled through the external auditory meatus to go and set the tympanic membrane into vibration, which is a mechanical vibration. So airwaves have energy, has been transmitted into a mechanical energy where the membrane is basically vibrating, which is transmitting this vibration to ossicles, which is again a mechanical type of energy, to the foot plate of stipes, which will transmit this mechanical energy into a fluid environment. So you would generate fluid waves. This mechanical energy or mechanical vibrations are going to be converted into fluid waves. And these fluid waves then will pass from the perilymph in the scala vestibuli into the scala media, where they are going to cause these basilar membrane to vibrate and the hair cells to get depolarized, which will result in an electrical stimulus going to the central nervous system. So this is how your normal mechanism of hearing is working. The cochlear duct, which is also called scala media, has this sensory organ, which is called the organ of Corti. And this organ of Corti is responsible primarily for the sensations of these sound waves to be detected and converted into electrical signals which can be perceived by the brain. In this organ of Corti, we have these hair cells which have these flexible stereocilia which beat, and beating of these stereocilia will result in depolarization of these cells, resulting in a neurotransmitter release, which usually in the case of the central nervous system, the excitatory neurotransmitter is glutamate. Okay, so unless somebody tells you anything else, in central nervous system, the excitatory neurotransmitter is glutamate. The initial processing of pitch and loudness and duration of sound is basically happening in the cochlea. And then localization and intensity of sound is basically assessed in the parts of the central nervous system. For you at this point, just remember there is a superior olivary nuclear complex which is present in the pons, which is important for localization and intensity of the sound. So we will talk about it when we are going to talk about the pathways, but for this point of time, just for an introduction, you should remember that there is a superior olivary nuclear complex in the pons which is responsible for localization and intensity of the sound. Okay, now you can see this is a frequency range chart, and you can find out that the humans are able to hear the sound between 20 decibels and about 20,000 decibels. But if you see the bats, they are basically going to hear the sound between 10,000 decibels and 100,000 decibels, so they can hear very high frequency sounds. Compare this to mice also, you would have, they can also hear a very high frequency sound because they can hear up to, 100,000 decibels, 100,000 hertz. This is the diagram which is showing you the same thing which we have been saying again and again, so I'm not going to diverge back into how the mechanism of hearing starts to work. So now we are going to talk about the different parts of the ear and what is their primary function. The first one is the outer ear, and the outer ear is primarily what it is doing is funneling the sound waves from the outside through this small canal which is called the external auditory meatus to go and hit the tympanic membrane. So the reason why it is doing it is because it is increasing the sensitivity to sound. Because sound is all around. It has to be funneled in, and as it is going to be funneled in, sometimes the sound is coming from the top, sometimes it is coming from the bottom. Your auricles are made in a way that they have curvatures so that it can make the sound waves to come and hit and reflect in different ways and then funneled into the ear so that you can detect from where the sound is coming, which intensity of the sound is coming. So all this is basically increasing the sensitivity of the sound. So your ear canal is able to boost the sound by funneling up to 15 to 18 decibels. And it can increase the frequency of the sound by 3 kilohertz, that means 3000 hertz. The sound could be amplified because of it is being funneled through the external ear. The other thing is that external ear has hair follicles, so it is going to make the tympanic membrane where this sound wave is going to hit, humid, and also the temperature would be maintained, so it would not be very swollen, or it would not be inflamed because of it is inside your ears. So because of those hair cells and the temperature control, the tympanic membrane remains healthy, and that would allow these sound waves to come in funnel through the external ear into the and hit the tympanic membrane. So two functions. One is that it is maintaining the tympanic membrane healthy. The other is that it is increasing the sensitivity of the sound by 15 to 16 decibels in terms of loudness, and about 3 kilohertz in terms of frequency of the sound. Then we have the middle ear, and in the middle ear, the tympanic membrane is at the partition between the external ear and the inner ear, and the middle ear. And the other thing which is there in the middle ear are these ossicles, the valgus, the incus, and the stapes. And there are two particular reflexes which the middle ear does, and these are the two major functions of the middle ear. One is the attenuation reflex, and the other is the impedance matching. So this is the primary function of the middle ear. And if you want to remember it in short, these attenuation reflex and impedance matching, although these look very fancy words, but actually what it is dampening and amplification. So the middle ear is a dampener as well as an amplifier. So attenuation reflex is dampening, and impedance matching is basically amplification. So in simple two words, if you want to remember, you don't want to get confused with what is impedance matching, and so you should remember impedance matching is amplification, and the attenuation reflex is basically dampening. Now, what is attenuation reflex? What happens is that if you hear very loud sound, what happens? You move your head towards that side, or you get shocked. What part of the brain is basically you have learned in neuroscience, which is important in terms of waking, or which keeps your consciousness? Reticular formation. So there is in your brainstem reticular formation. So whenever you are going to hear a very loud sound, your hair cells will get stimulated. The cochlear nerve is going to go and stimulate your pons and medulla, and you have this big reticular formation which is there in pons and medulla. It would get stimulated. This would stimulate two nuclei. One, the nucleus of the motor nucleus of trigeminal, which is also in pons, and it would also stimulate the motor nucleus of facial, which is also in pons. So this reticular formation is basically causing what? Bilaterally, it is going to stimulate both the motor nucleus of trigeminal and the motor nucleus of facial. They are going to, motor nucleus of trigeminal through the mandibular nerve is going to supply the tensor tympani muscle, which is going to pull the malleus inwards. And similarly, the facial nerve is going to supply the muscles, which is called the stapedius muscle, which will put the foot plate of stapes out of the oval window. This will make this ossicular apparatus very rigid, and it will not allow it to vibrate. So that would save your inner hair cells. And there would be a latent period of 40 to 80 seconds before this reflex starts. So it would not start the minute you hear the loud sound, because it has to go to the reticular formation. Reticular formation needs to get activated. It would activate bilaterally both, your nuclei of the facial and mandibular nerve, causing the contraction of tensor tympani and the stapedius muscle, which will result in dampening. So you would be able to save your inner hair cells from the loud sound. But there is a latent period of 40 to 80 seconds. This is called attenuation reflex. Now, the other one, which is called the impedance matching. Now, impedance matching, as we said before, that impedance means obstruction. So, when the sound waves are traveling in the air, they have less obstruction as compared to when they are going to travel in the fluid. So, because your sound waves initially travel in the air and then they hit the tympanic membrane, vibrations are set in the tympanic membrane, which causes the ossicles to vibrate, and then the foot plate of stapes is going to cause the vibrations to be transmitted into the fluid, which is the perinerve. So that the impedance or obstruction to these fluid waves would be much more higher as compared to the obstruction to the air waves which are traveling in the air. So you need to match that obstruction which is in the air to what they are going to get the obstruction in the fluid. So it's a very interesting mechanism where ossicles, because they are lever systems, so they would increase the force in this in the sound waves by 1.3 times. And if you see the tympanic membrane, the size of the tympanic membrane is around 55 millimeter square, the circumference. And if you see the foot plate of stapes, which is going into the oval window, the size is 3.2 millimeter square. So the force which is applied to the tympanic membrane versus that force is concentrated into a very small area, very large area and to a very small area. Now you could understand why your tympanic membrane has to be tilted, because it's so big, you need to put a big thing in a place and you have less space. So you put it in a tilted manner so that you can allow this large circumference. So when the sound waves are hitting on the tympanic membrane, they would be concentrated into a smaller place, which is 17 times smaller. So the force which was applied to the tympanic membrane has now increased 17 times. So now you multiply this 17 with 1.3, it will make you around 20 times more than what was there in the air. So you are matching the impedance in the air versus in the fluid by putting up this structure within the middle ear. One is that the tympanic membrane is bigger to the size of 17 times larger than the size of the footplate of the stipes. And the other thing which is important is that you have these ossicles which acts as a lever, and they would increase the force by 1.3 times. And if you multiply this would increase it by 22 times. Okay? So this is the impedance matching. So these are the two functions. So this is amplification actually. You can name it fancily, impedance matching, but it's basically an amplifier. That's all what is happening. Okay? Reasons of amplification we have already talked about. Okay. Next, we are going to go into internal ear. And this is the place where we are going to have the majority of our discussion today, the internal ear and the pattern. So, the internal ear, if you see that this footplate of stipes is going to vibrate against the oval window, is going to set up the vibrations in the scala vestibuli, which is the upper part of the cochlea. And then this scala vestibuli's vibration will be transmitted into the scala media, where you have the basilar membrane. And if this vibration which is there matches the frequency, it would set the basilar membrane, that part of the basilar membrane, to vibrate. If not, these vibrations will pass through the helicotrema into the scala tympani, and then ultimately will cause the bulging of the tympanic membrane, which is the secondary tympanic membrane here, which is covering the round window. Okay? So this is the normal mechanisms. Now let's look at the scala media in detail. So what you are going to see in the scala media, there is a membrane on the top which is called the vestibular membrane or Reisner's membrane, anything you can call it. Above this is the scala vestibuli. You have the basilar membrane here. Below this you have the scala tympani. Both scala vestibuli and scala tympani has perilymph, which is like CSF, high in sodium. And then you have the scala media, which is produced by this structure here laterally, which is called the stria vascularis. And stria vascularis actively pump potassium into the scala media. Now, the basilar membrane is the place where you have these hair cells. You have these inner hair cells, which has one row of cells, and then you have these rods of corti. See these rods of corti. In between there is this tunnel of corti. Then you have these 3 rows of outer hair cells, and then there is a membrane which is connecting all the tops the top layer of hair cells. This is called the reticular lamina or reticular membrane. Some people call it reticular lamina or membrane, which is going all the way like this. Above what is this gelatinous structure, or which is called the tectorial membrane. Most of your cochlear nerve fibers, 95% of them, they are going to synapse here on the base of the inner hair cells. Only 5 to 10% are going to go and synapse to the base of the outer hair cells. So the major hearing cells which are responsible for your hearing are the inner hair cells. Outer hair cells are for modulation, and we will talk about what type of modulation they do. Again, this is showing you the same structure. This is the inner hair cells, these are the outer hair cells, this is the tectorial membrane. This is the basal membrane, and you can see that 95% of the cochlear nerve fibers which are coming from the ganglion are going to supply the inner hair cells. 5 to 10% are going to supply the outer hair cells. This we have already talked about. Okay, now this is again important. So if we see the basement, the basal membrane on which the hair cells are lying, the inner hair cells we are talking about, it is narrower near the base and it is very tight near the base of the cochlea. It is narrower and tighter. As it goes towards the apex, it becomes wider and floppy. So because it is tighter near the base, the high frequency sounds are going to make the part of the basilar membrane which is near the base to vibrate, while low frequency sounds are going to cause the basilar membrane near the apex to vibrate. So this is the way cochlea is going to identify if you are hearing a low frequency sound or high frequency sound. Low frequency sound near the apex, high frequency sound near the base. So this is an important concept which you need to have in mind, that we can identify the frequency of the sound at the level of cochlea by just seeing which part of the basilar membrane is vibrating. If the basilar membrane near the base is vibrating, it's a high frequency sound. And if it is near the apex, it is a low frequency sound. So you see this diagram, you see number 1, 16 hertz, 1600 hertz. Number 7, which is near the helicotrema or near the apex, is only 25 hertz. So low frequency sounds are going to be near the apex of the cochlea. The basilar membrane, the part of the basilar membrane near the apex is for the low frequency sound. Near the base, a high frequency sound. Now, the other thing which can also be assessed by the cochlea is the loudness of the sound. How the number of hair cells which in that area, if they get stimulated, is going to determine the loudness of sound. If more the number of hair cells get stimulated, they would result in more loudness. If there is less number of hair cells stimulated, less loudness. So both loudness and frequency can be assessed by the preliminary assessment of loudness, and the frequency is done at the level of cochlea. Okay? You understand everybody?
Yes.
Okay. So this is again showing you the same. You see that the basilar membrane near the base is narrower and it is tighter, and as you go towards the apex, it is becoming wider and it is floppy. So you would have base, high frequency, apex, low frequency sound. Now we're coming to the how these hair cells actually get excited or depolarized. So what you are going to see, these hair cells, especially we are now at the moment talking about the inner hair cells, and then we will talk about what the outer hair cells do. So if you see the inner hair cells, you have these specialized, upper ends which are called stereocilia, and then they have a very long one, projection which is called gynoecilia. In between them, you have these proteins bridges. Can you see these protein bridges?
Yes.
These protein bridges are called TP links. TP links. It's not written here, but it's called TP links. Now, whenever your basilar membrane, when your sound waves are coming through the foot wave plate of stapes, they pass through the scala vestibuli into the scala media, and now the frequency has matched and your basilar membrane is vibrating. As the basilar membrane is going to vibrate, it is going to allow this endonymph, because the basilar membrane is, and the hair cells on the top of it are covered by the tectorial membrane. When it starts to vibrate, it would allow the endolymph to pass between the tectorial membrane and the hair cells. And if your you're basically if your basic stereocilia are vibrating towards the gynoecilia, what would happen is that these TP links will open, and because your endolymph, which is high in potassium, is entering here, it would enter into the cell, and this will result in depolarization of the cells. Once the cells are depolarized, there are calcium channels which are on the side. They would open, calcium will come in, this will bind to these vesicles which are containing the neurotransmitter, the glutamine, and result in the release of these glutamine into the synaptic cleft, resulting in the action potential in your cochlear nerve, which will go and stimulate the cochlear nuclei and all those type of things. Okay? So again, as the endolymph is going to gush in, it is going to cause the stereocilia to move towards gynoecilia, opening the TP links, which are protein-built bridges, and this will allow the potassium to go in, resulting in depolarization of these hair cells, the innate hair cells. And then the calcium gushes in, and as the calcium comes in, it is going to bind to these vesicles, which are containing neurotransmitters, and result in the release of this neurotransmitter in the synaptic cleft. And mostly this neurotransmitter is glutamate. Aspartate can also do it, but usually it is glutamate containing neurons. Now, yeah.
I just wanted to know how does endolymph get inside the space between the
Yeah, because it goes down, as the membrane is vibrating, so it's moving like this. So there is a space which is generated between the tectorial membrane, and it is also moving the stereocilia. So stereocilia is moving against the tectorial membrane, and there is also space generated in which the endolymph is coming in. And that endolymph is because it is potassium rich, so it's going to cause it depolarizes itself, opens the calcium channel. Now, there's something interesting which happens with now imagine that these are not inner hair cells, these are outer hair cells, okay? So something else is happening. Outer hair cells are supplied by cochlear nerve, but these are usually not afferent fibers. These are usually efferent fibers. And they are releasing a neurotransmitter which is called acetylcholine. They are not releasing glutamine. These efferent fibers are basically releasing what? Acetylcholine. Now, on the hair cells of the outer hair cells, we have something called some proteins which are called Preston molecules here. They are basically proteins here, and they are called Preston molecules. They are called T-I-N. Okay, Preston molecules. Now, when the acetylcholine is going to be released here, it is going to cause a hyperpolarization of these cells. And these Preston molecules, which are proteins, press means compressed, but they get relaxed. So when the cell get hyperpolarized, the outer hair cells get hyperpolarized, the Preston molecules are going to relax. This will push the hair cells down. As the hair cells are going to be pushed down, outer hair cells are 3 rows of them. And if they are pushed down, they would push the basilar membrane down. They push the basilar membrane down, they would not allow the hair cells, the inner hair cells, to move against the tectorial membrane, and there would be, downward bowing of the basilar membrane. So this is a protective mechanism against loud sounds. So when you hear loud sounds, there's an efferent going into the outer hair cells, which is going to cause the hair cells to push the basilar membrane down, or downward bowing of the basilar membrane, which will cause the dampening or protection of the inner hair cells. So one mechanism is this. The other thing which can happen, there could be another neurotransmitter which can be released here, and I don't remember the neurotransmitter which can be released here, which will result in depolarization of the outer hair cells. And then if they get depolarized, then these Preston molecules are going to contract. And this is going to make the upward bowing of the basilar membrane. So this is done for increasing the sensitivity of the sound. So if you want to have that sound to be more sensitive, you want to have more sensitivity, there would be upward bowing of the basilar membrane, which is going to cause more sensitivity to the sound. So both mechanisms are there. So you can see this is like a fine-tuning mechanism. So you want to fine-tune, make it more sensitive, or you want to decrease it or dampen it. So this is the mechanism which the outer hair cells do. So if even your outer hair cells are all dead, you still would be able to hear, because it's the inner hair cells which are responsible for hearing. Outer hair cells are either dampening or increasing the sensitivity.
Just a question.
It's a volume, thing. So it's just like a volume tap. You want to increase or you want to decrease the sound. Yeah.
When the basilar membrane goes down, wouldn't the endolymph flow to the inner hair cells?
No, but because one of the important thing for stereo to beat is that it moves against the tectorial membrane. There is, if it is going to go down, it's not hitting the tectorial membrane at all. Even you have endolymph, the TP links will not open because there is no cilia movement. Yeah.
So in the case of the diameter of the outer hair cells, would that cause like hyperacousis?
Sorry?
Would that also cause like hyperacousis as well?
It's just, what can I say? This is just a small volume tap which you have here. It doesn't cause hyperacusis. Okay. So this we have already done. I'm not going to talk about this. Now we are coming to the auditory pathway. I would discuss this auditory pathway in detail, and I made this diagram which is going to be used for discussion. But I would strongly advise that there is a video by Ninja Nerd on cochlear pathway. It's a good one. You can hear it. Even though it is 40 minutes long, but you can hear it at 1.5, it would finish in 20 minutes, something like that. But it's a good one, and it's something on cochlear nerve pathway, and that's a good one to remember. But it's derived from the same concept. So you have your cochlear nerve basically is arising from the spinal ganglion, especially 90% of your inner hair cells are basically bringing, it's a pseudo-unipolar type of neurons there in the spiral ganglion. Peripheral process is going to inner hair cells. Your central processes are going towards the pons where the cochlear nuclei are located. So this is going to become a cochlear nerve, all these fibers, and then they would pass through the internal acoustic meatus to the junction of pons and medulla, where you have cochlear nuclei. And usually you have two sets of cochlear nuclei. One is posterior to the inferior cerebellar peduncle, which is called the posterior or dorsal cochlear nuclear complex. And then you have an anterior cochlear nuclear complex, which is anterior to the inferior cerebellar peduncle. Now, the posterior one is easy because it is not further divided. The anterior one is further divided into two groups, which we are going to talk in a minute. So the dorsal cochlear nucleus is going to, give rise to the second order neurons. These second order neurons are going to cross the midline. They are called, when they cross the midline, they are called dorsal acoustic stria. Oh, yeah.
This is the brainstem, right?
Yeah, brainstem. So when they cross the midline in the pons, they are called dorsal acoustic stria, and then they reach this nucleus, which is called the nucleus of lateral lemniscus. And from the nucleus of lateral lemniscus, they would join other fibers to become the lateral lemniscus, and they will go to the inferior colliculus, and from inferior colliculus to the medial geniculate body, and then to the superior temporal gyrus, where the auditory cortex is. But you need to see that there is, communication with the nucleus of lateral lemniscus from one side to the other. Similarly, there is a communication between the inferior colliculus on both sides. So hearing is bilateral. So it's not that if you damage one side nucleus, you would have complete hearing loss, because the other side is there, and you would have communication from their side. The second group of nuclei, which are the ventral group of the cochlear nuclei, you divide them into ventroposterior and entroventral group of cochlear nuclei. Now, why they named it entroventral and ventroposterior nucleus, don't ask me. But this is how it is. So this ventroposterior nucleus, cochlear nucleus, is going to give intermediate auditory stria, which is going to cross the midline. It is going to go all the way up with the lateral lemniscus to the medial, to the inferior colliculus, and from inferior colliculus to the medial geniculate body, and then to the cortex. The one which is interesting is this one, which is called the anterior-ventral cochlear nucleus. Now, the fibers which are going to cross the midline after synapsing here, they are partly going to go to the ipsilateral side, partly they are going to go to the contralateral side. And these fibers are either called anterior auditory stria or trapezoid body. These are the fibers which are called trapezoid body. And they are going to this complex, which is very important, which is called superior olivary nucleus complex. These fibers, which are coming from the anterior-ventral cochlear nucleus, they are crossing as well remaining on the ipsilateral side. They are going to go to the superior olivary nuclear complex. And there are two parts of this superior olivary nuclear complex. One, it is called medial superior olivary nuclear complex. The other one is called lateral superior olivary complex. Okay? But they are going bilaterally. They are going this way as well as to the other side, superior olivary nuclear complex. Both sides, they are basically supplying. MSO, which is the medial superior, this is not MSA, but MSO, the medial superior olivary nucleus, is important for lateralization, that from which side the sound is coming. So that's why it is going on both sides. So, but the action potential if you are hearing from the right ear, the action potential to the medial superior, the right medial superior olivary nucleus will reach first, then the other side. So that's how the ear would determine that the sound is coming from the right side or the left side. So remember, MSO, the medial superior olivary nuclear complex, is important for lateralization, from which side the sound is coming. Yeah.
But then wouldn't the sound be better to both ears?
Yeah, it is coming from both ears. But the action potential generated here would reach the MSO of this side before the other side MSO is going to reach. So that difference of milliseconds of action potential reaching to the two MSOs will define from which side the sound is coming. The lateral superior olivary complex is important for deciding the intensity. If the sound has higher intensity coming from this side, so sound is coming from this side, or this side, the intensity of the sound would be determined by the LSO. Because if it is the depolarization of one side of LSO has been done, the other side would get inhibited. So that would actually determine the intensity of the sound. Yes?
So it would be the same concept if one side is depolarized first, and it's from both ears.
Yes. So intensity of the sound is basically, or the timing of intensity of the sound, relative intensity of the sound, that the higher intensity is from here or the other, is basically decided by the LSO. Now, after doing all this, the ventral acoustic stria, going to the superior olivary complexes, it will go all the way up, forming the part of the lateral lemniscus to the inferior colliculus, and from inferior colliculus to the medial geniculate body, and then from medial geniculate body to the auditory cortex. So these are the two important concepts, lateralization and relative intensity, which you should know. Again, this is a picture from Ninja Nerd, and you can look at it. So this is basically showing you the lateralization. If the sound wave is generating here, it will reach the MSO on the right side before the left ear is going to send its impulses to the other side. So it would be a little bit of delay, and that actually tells you the lateralization. And the intensity here, the LSO, if it is excited on this side, the other side will get inhibited, and that actually tells you the intensity of the sound, which is relative intensity, on the right or the left, which is higher. Yeah.
Okay, so if one side is depolarized, other side is inhibited, and then this other ear will activate this contralateral side.
Yeah.
Wouldn't that also inhibit the?
Yeah. So you would know that in terms of localization of the sound, in terms of timing, from where it is coming, that is MSO's work. And if it is, in terms of intensity, relative intensity, you will find out that the sound is coming from this side or that side, because the LSO, where you would have high intensity, would be activated on that side and inhibited on the other side.
But wouldn't the other ear have the LSO on the contralateral side activated, but then?
No, it would not, because the other side is already inhibited by this side.
So it's that it got inhibited first, so it wouldn't get activated.
No.
Okay.
Okay, now we're coming to the last piece, which is the deafness and tests of deafness. So inability to hear is deafness, and we have two types of deafness. One is the conductive deafness, which is due to the impairment in the transmission of sound into cochlea. Causes could be foreign body, wax in the external ear, otitis media, which damages the tympanic membrane, or ossicles are not moving. There is a pathological fixation which happens in old age, in some people, which is called osteosclerosis, where you would have the foot plate of stipes fibrosed, so it is not moving against the oval window, so that is called osteosclerosis. You can also have nerve deafness or sensorineural deafness because of degeneration, hereditary causes, some drugs like quinine, which is used for malaria, can damage, inner hair cells. Acoustic trauma, which can damage the hair cells. Meningitis infection going into the inner ear, damaging the hair cells can cause. Acoustic neuroma, you can have this tumor of the cochlear nerve, and this is it's also called schwannomas sometimes. It will compress the nerve, and as it's going to compress the nerve, you are unable to hear. So acoustic neuromas can also happen. There are tests which we do. One is the Rennie's test. Rennie's test, what you do, you take a tuning fork, you hit it, and then you put it near your ear, and then you put it on the back, on your mastoid, bone. So you're testing the air conduction versus the bone conduction. If you have a conductive deafness, you would have normally what you should have air conduction better than bone conduction. But if you have a conductive deafness, you would have bone conduction better than air conduction. So this is basically the principle of the Rennie's test. If the person has the nerve deafness, it doesn't matter. Neither the air conduction and bone conduction will all be similar. So usually Rennie's test is done for identifying conductive deafness. Now, if you are doing the Weber's test, which is shown here, what you do is basically put up the tuning fork, hit the tuning fork, put it in the center on the center of your vertex of the head. Normally, the sound waves would travel equally on both sides. Now, what would happen if you have a conductive deafness on this side? Your sound, because the background sound is gone, so you would hear in the affected ear better than the unaffected ear. So in the Weber's, if you have a conductive deafness in a particular ear, you would have a better hearing in that affected ear versus non-affected ear. You understand? Because the background sound is gone. Your conductive deafness has stopped your background sound. So you are hearing better on the affected side versus the unaffected side. But if you have a sensory neural deafness and you are doing the Weber's, you would hear better in the unaffected ear, and you would not hear better in the affected ear because your cochlea is gone. So you are not able to basically hear in the affected ear, but you would hear better in the unaffected ear.
Okay, in this specific case of conductive hearing, you notice the conduction would be more in the affected
You would hear better in the affected ear.
But for the sensory neural, the normal side would hear like it
Better.
normally does.
Yeah, normally does or better than the unaffected. You are comparing better, where you would say to the patient where you are hearing better, in this ear or that ear. You are not basically, you are asking the patient where you hear the sound, this side or that side. So you are determining based on that interpretation where the problem is. Is it the sensory neural problem or is it the conductive problem?
But then in the case of conductive, the affected ear would be able to hear better than a normal ear would.
But there is no comparison with the normal ear. Here you are comparing with the affected ear. Okay? Okay, last slide. So we have something which is called presbycusis, which is the gradual deterioration by age of the spiral organ of porti. You lose hair cells as you age, and based on that, your hearing is affected. 1 in 3 older adults over the age of 65 has hearing loss, and about half of the people over 75 have hearing loss. Somewhat, you will not have a complete hearing loss, but your hearing gradually declines as you age. Like you have presbycusis, when your lens become much more less stretchable, so you cannot accommodate. So you start to have reading glasses as you age. Similarly, you have presbycusis, which happens when you age and your hair cells are lost over the period of time, and you start to have less and less of your hearing ability as you age.